Hi there, welcome to this video. This is 3.4, which is about prokaryotic cells. So first of all, what does prokaryote mean? So it translates as before the nucleus. So prokaryotes evolved before the nucleus evolved. So the kind of definition of what they are is that they have no membrane-bound organelles. So if you imagine something like an animal cell, and you would have inside the animal cell, you'd have like the endoplasmic reticulum, for example, something like this. That outline that I've drawn there is all membrane, so it's a membrane-bound organelle. Even a mitochondria has got an outer membrane and then a folded inner membrane. So it's got a membrane around the edge, so it's membrane-bound organelle. So a prokaryotic cell would not have any of those. Okay, so we say that prokaryotic cells have no membrane-bound organelles. So no nucleus, which is the key one, but also no endoplasmic reticulum, no Golgi, no mitochondria, chloroplasts, no vesicles. Okay, none of those organelles or subcellular structures that your eukaryotes have. Okay, so they're much more simple, much simpler. They do obviously still have DNA, and they have all the enzymes that are doing all the jobs, catalyzing reactions in all the other organelles. But those enzymes and that DNA is just free in the cytoplasm. It's not inside these little compartments called organelles. So the DNA loop, because the DNA is a loop, not in chloroplast. Oh, sorry, not in chromosomes. The DNA loop is free in the cytoplasm. Okay, so let's have a look at drawing a prokaryotic cell. So the classic shape is like the almost tadpole look, but obviously, different bacteria in reality are different shapes. So we've got this inner membrane, which we call a cell membrane. And then almost like a plant cell, we've got another layer, which is called the cell wall. Now, sometimes, but not always, we've then got a third layer around the outside, which is called a capsule. Then we've got this classic kind of tail, which we call a flagella. There for movement. As I said before, the DNA is a loop. It's a bit squiggly, but it join your ends up because it is one big loop, but it's not in a nucleus. Then we got these extra little loops of DNA, which we call plasmids, much smaller. Then the inner membrane has these little folds in it called mesosomes. Now, the function of those mesosomes is thought to be to do with providing a surface for the enzymes for things like respiration, because it hasn't got all of those membranes inside the mitochondria for the enzymes to attach to catalyze respiration. It's thought that maybe those little infolds of the cell membrane called mesosomes are the kind of anchor for those enzymes to catalyze those reactions. Okay. So let's have a look at what all of those different parts actually do. So first of all, the cell membrane, just like in eukaryotic cells, the cell membrane can control what enters and what leaves the cell, what can go in and out of the cell. Okay. So let's write that down. Let's put them on the next page. Oh, sorry, I should also say that you know how the rough endoplasmic reticulum provided a membrane for the ribosomes. we still do need ribosomes, still need to make proteins, but again, they're just loose in the cell, not attached to a membrane. They haven't got a special membrane for them. Okay. Just checking if there's anything else. Oh, the last thing is just these little pili. So they have these little bits sticking out from the edge here, and they can just be used to attach onto other surfaces. Okay, so let's go through the functions of each part now. So we said already, let's make this a bit bigger. We said already that the cell membrane, just like any cell membrane, controls what enters and leaves the cell. The cell wall has the same job as all cell walls, but it's made of something different to what you would make it from in a plant. So it's made of a glycoprotein. So the glyco part means carbohydrate, protein obviously means protein. So it's a glycoprotein cell wall. In plants, it's a cellulose cell wall. So just remember that it's made of something different. And the function of a cell wall is just a support, a structural role. Then we said that sometimes we have a capsule, but not always. Let's put sometimes. And the role of the capsule is actually a few roles. So the first thing that the capsule might do is prevent it from dehydrating. The other thing it can do is it can provide some kind of protection from kind of other cells that might try to sort of attack it. So, for example, if a bacteria got into your body, your immune system might try and attack it. So the capsule makes it harder for your immune system to kill it. So first thing is it can prevent dehydration. And the second thing is it can be protection, e.g. from your immune system. Okay, so that's our three walls on the edge. Then the other things we've got on the outside is we've got the flagella, which is the kind of tail-like structure that's just for movement. Some bacteria don't have flagella, some have more than one flagella, but that's what it's for. And then we've got these little pili around the outside, which we've mentioned briefly. They're like little hairs sticking out from the edge of the cell, and they allow it to stick onto other surfaces, to attach onto other surfaces. Now, you might have heard of how sometimes one bacterial cell can actually form a little transfer tube across to another bacterial cell, and it can make a copy of a plasmid and send the copy into the other cell. And so, if the plasmid coded for example, antibiotic resistance, it can make the bacteria next to it antibiotic resistant as well. And that actually happens with using these pili as well. So it's involved in the transfer of genetic material between the cells. Okay, so that's the outside of the cell. Now let's look into the middle of the cell. So obviously we've got going in from the membrane, we've got these little folded bits from the cell membrane, and they're called mesosomes. And they're literally little inward folds of the cell membrane or of the plasma membrane, and it's not 100% sure what they think that they are. They might be just kind of they might be involved in cellular processes, they might be attachment for enzymes, etc. And that's kind of the most likely view. There are actually some people that think that they're not, they don't really even exist, that they're just like mistakes on the microscope almost, but there is evidence that they do exist, I think, nowadays. So mesosomes, they're inward folds of the cell membrane. And I'll put possibly for respiration enzymes to attach to. So they would normally be attached to that folded membrane inside the mitochondria. So perhaps these are just providing a surface for those. Then we've got our main DNA. So the main DNA is not in chromosomes, okay? So we're used to DNA being folded into chromosomes, but in bacteria, the DNA is not folded into a chromosome, and it's also not in a nucleus. So it's a circular piece, okay? Now, our DNA is wrapped around proteins called histones, and that's how it folds into that specific 3D shape to make the chromosome shape. So this is not associated with any histones because it doesn't need to fold up, okay? And it's free in cytoplasm. And we've got these extra bits of DNA, which are called the plasmids. So these are very small loops of DNA. And they often, not always, but they often code for antibiotic resistance. And that's what I was saying. Sometimes they can transfer them from one bacterial cell to another bacterial cell. Not all prokaryotes have plasmids. Some might have one, some might have many. It really varies. Okay. We said they have ribosomes, which are obviously for protein synthesis. Come across those lots of times, haven't we? The ribosomes here as well, it's worth saying now that they're 70S. In eukaryotes, they're 80S. That's a different density. That's how they measure the ribosomes. And I think that's it. Yeah, okay. So that's our structure of prokaryotic cells. So make sure you're not going to have to draw it, but make sure you can label a prokaryotic cell, and make sure you know the function of them as well. Okay, and that's the end of that split point.